my dear little champions, I welcome you all to score 360 out of 360 in NEET Bio within 250 days. And today we are going to talk about transgenic animals, GA, AC and biopiracy, which is the last topic of this particular chapter, that is biotechnology and its application. Now, what did we cover in the previous session, my dear children? We spoke about humulin, the gene therapy and medical diagnosis. In everything, we saw one thing is common, that is what? Biotechnology. We saw how it was used for different purposes. Now, if you want the PDF of the same, you can join our WhatsApp channel, okay? And if you want to see the whole videos that we have covered until now, my dear children, you can go to the playlist which has been created for NEET. Okay, go according to the number, because for every video we have given the numberings, so it will be easy for you to understand. Now, the number of questions that have come from this chapter is 4 into 4, 16 marks from this particular chapter, something that is really good. Imagine studying a very limited thing and getting great marks for it, because this chapter is not very huge, it is a very small chapter, and if you are able to score really good by studying just a very little amount of thing, then it's really good. Why are you ignoring this chapter? Now, going ahead, I remember I gave you a homework. Let's see who all got it correct. So, champions, the question was, the term humulin is used for what? Why do we use the term humulin? Basically, humulin stands for human insulin, my dear children, and it was being produced not in the humans but in a transgenic organism, that is E. coli. So, in E. coli, the gene for making alpha peptide, okay, or basically, I won't write alpha peptide, I'll write A peptide, this is a peptide, and B peptide. This is again another peptide. When both of them, they come together, you get insulin. So, basically a gene which was making this particular peptide and a gene which was making this particular peptide was being put inside E. coli's plasmid, and then E. coli was making this. Later A and B was taken out and then joined to form insulin. This is human insulin, okay, and Eli Lilly was the company which actually did it. Now, we studied about it in the previous session. It's a very easy question. So, your answer becomes what, children? Human insulin. Works? Now, going ahead with the next thing. So, transgenic animals, what do you mean by that? We have studied a lot on them, but what do you exactly mean by that? So, animals that have had their DNA manipulated to possess and express an extra gene are known as transgenic animals. For example, I have this cute little mice over here, and now this is the DNA that we have. Okay, and do you know our DNA and the mice DNA, it's almost very similar. Okay, so what we are doing is that we are cutting down a portion of their DNA which is coding for some protein. So, I am cutting out this portion that you see over here, which probably I'll mark it in red. Okay, I am cutting it out, and what do I get now? This is the gene that I get now, where I do not have the red portion. Now, I'll see what is that effect that is being that feature that is getting affected because I do not have this particular gene in the organism now. So, this manipulation that I am doing in their DNA, which is helping me understand the function of this particular gene. I am seeing if this particular gene is not there, what is that particular feature that is going to get affected. Okay, I am studying with the help of them about the features of physiology of human beings as well, because we share a lot of details together. Same. Okay, so basically I am manipulating the DNA, or probably I can do one more thing, that is I can insert one gene and see what does it do, what is that product that is being produced. I cannot insert gene inside human beings, because obviously we will not test on human beings ourselves. Instead we test on animals and stuff like that, which is equally very wrong. There are ethical issues being arisen, but still people definitely do it for let's say knowing about different diseases, for bringing out a cure and things like that. So, anywhere where you are manipulating the genetic material of some other organism for your own use, it will be known as transgenic animals. The animals that are being produced like that, they are known as transgenic animals. So, animals that have had their DNA manipulated to possess and express an extra gene are known as transgenic animals. So, over here they are saying when I am putting a gene and then it's showing up this particular organism becomes a transgenic animal. Now transgenic rats, rabbits, pigs, sheep, cows and fish have been produced. You have to remember the names. Rats, rabbits, pigs, sheep and cows and fish are being produced, although 95% of all existing transgenic animals are mice. The question has come, which is the most commonly used transgenic animal, children? It is mice. 95% of our experiments are done on mice itself because of the very close similarity that we and they have in the case of the genetic material. Okay, so rats, rabbits, pigs, sheep, cows and fish. Not think that's a very little simple thing, okay, and it will be really easy for you to remember during the year's exam. I'll suggest you to have a big clarity while reading this. Questions have come, they have asked which among them is not usually used as a transgenic animal. So you should be knowing which of them is not. So usually we remember all this thing but not the fish. So fish is important. Okay. Now going ahead, let's see what is the use of transgenic animals in different things. First, to understand normal physiology and development. So this is your NCERT. I'm going to read your NCERT and then explain things to you. Okay. So transgenic animals can be specifically designed to allow the study of how genes are regulated and how they affect the normal functions of the body and its development. Example, study of complex factors involved in the growth such as insulin-like growth factor by introducing genes from other species that alter the formation of this factor and studying the biological effect that results. Information is obtained about the biological role of the factor in the body. So, our hero of the story is insulin-like growth factor. And we are assuming that's already there in mice. The gene for this particular factor is already there in mice. Okay. Now what are we doing? We are doing a really bad thing. We are going to introduce certain genes, okay, certain genes that is going to alter the expression, of insulin-like growth factor, my dear children. So now what will happen in the case of mice which had insulin-like growth factor, they were releasing it normally. Now I have put some factor which is going to alter the expression of this and it is not being produced now. Now the mice is something different. This is a transgenic mice. Now I will see what will happen to these mice if this particular factor is not there. When I am doing this, I am able to understand, okay, so A function is not happening, B function is not happening, C function is not happening. In the case of this new transgenic mice, that means probably insulin-like growth factor were responsible for all these functions. So I am getting to know the role of one particular gene or one particular protein in my body as well with the help of this particular experiment. So to understand the normal physiology, normal work process of how things actually work and the development, we are using transgenic animals. Okay, works. So this is one thing. So to understand the role of one particular gene, I'll make it easy for you to understand the role of one particular gene. Works. Now next thing, study of a disease. Let's see how. So many transgenic animals are designed to increase our understanding of how genes contribute to the development of disease. These are specifically made to serve as models for human diseases so that investigation of new treatment for diseases is made possible. Today transgenic models exist for human diseases like, okay, so basically what is happening over here, I am using this particular organism. For example, let's take QT mice only over here. Okay, and I am going to use it as a model so that I am putting certain different type of genes into it. I am giving treatments to it such that I understand that which treatment could finally work. Now the same example that I gave you, that is insulin-like growth factor, if I am altering it, what is a disease that is being caused in the mice, children? It is known as diabetes. So I'll get to know that diabetes is being caused of that particular thing, and hence I should be giving the organism or anyone who is affected by diabetes a dose of insulin or something like that. So basically it is helping me understand the disease as well, and also different treatment methods that can be put on them, okay, or put on us later on in life. So with the help of this, there are so many diseases that have been there are so many transgenic models that have been made for understanding the role of these diseases. What are those diseases? Let's see. So cancer, cystic fibrosis, rheumatoid arthritis and Alzheimer's. You have to remember their names. What is it, children? You have cancer. So transgenic models were made, okay, such that I can put certain treatments on them and see whether it is helping to cure cancer or not. So cancer is there. Apart from that, cystic fibrosis is there. We studied in genetics about cystic fibrosis, right? Next thing is what? Rheumatoid arthritis. We studied in rheumatoid arthritis. We studied in human health and diseases about rheumatoid arthritis. It's an autoimmune disease. Rheumatoid arthritis, okay, wherein the joints and all, it starts paining way too much. That happens. And Alzheimer's, where you tend to forget a lot of things later on. So these are the things where transgenic models were used to study the disease very effectively. So studying the disease is also important. So you got to know the role of the gene, children. Then you got to know how we can use different treatment methods, or what actually causes the disease and what can I do to cure the particular disease as well. Okay. Now going ahead, biological products. For making, in the name suggests, you might be able to easily understand it. I want to make so many proteins, okay, which probably I cannot make in my body because I cannot extract things in my body. Obviously I am a living organism which has high importance in the society, right? So obviously things cannot be made worked on me, okay, because we do not use human beings as a transgenic animal. Even though mice should not, as I said, mice or any other organism should also not be used, but since they have the reproductive capability to increase the number very much, and they have also acted as pests, and they have lots of common features to us, which is why we use them. I am again saying it's not entirely correct, but this is how things work. So we are making use of these models for basically bringing out the products that we need, and probably it is not going to hamper their development as well, because they are in their body, we are just putting a gene and that gene is producing some product and we are taking that product from them. Okay, so let's see how it's not always killing, not always killing that is being involved, which is why I am telling you. Okay. This is something interesting. Let's read it once. So, medicines required to treat certain human diseases can contain biological products, but such products are often expensive to make. Transgenic animals that produce useful biological products can be created by the introduction of the portion of the DNA which codes for a particular product, such as human protein alpha antitrypsin used to treat emphysema. So, what are we doing, children? There is a disease which is known as emphysema. This is a respiratory disease, okay? This is a respiratory disease. So now for treating this disease, you need a protein which is known as human alpha antitrypsin, okay? Now, this is a protein, alpha antitrypsin is a protein, agree? And proteins are coded by genes, agree? So why not put this gene into an organism which can produce the same thing in their body, and then we can take it out. So basically human alpha antitrypsin is a protein which is very much important for the treatment of what? Emphysema. So that was being produced, okay, with the help of introduction of this gene into some transgenic organism, and then production of this thing which could be used to treat this particular disease. That is one thing that you have to remember, children. That is one example that you have to remember. Similarly, similar attempts are being made for treatment of phenylketonuria and cystic fibrosis. So there is one more example of a disease, that is phenylketonuria. Phenylketonuria, this is a disease, okay, or you call it phenylketonuria, PKU, and cystic fibrosis. We were making products for treating them as well. Okay. Apart from that, that's a very important example. You have to pay attention. So in the year 1997, the first transgenic cow, Rosie, produced human protein enriched milk, that is alpha-lactalbumin. What is it, children? Alpha-lactalbumin was being produced in a cow. 1997, a cow was being produced, transgenic cow, and the name of the cow was Rosie. What was it doing, children? It was making high protein milk. high protein milk, and this particular protein is basically human protein. Human protein. And what is it, children? What's the name of that? Alpha lactalbumin. It is alpha lactalbumin. So alpha antitrypsin is a protein that is used for emphysema treatment, and then you have alpha lactalbumin, which is a human milk protein found in human beings. Now if you are putting the gene, putting the gene which is producing this protein into the cow, okay, and calling it a transgenic cow, that is Rosie now, and it is producing the same type of milk which is having the same type of protein, then why not use it? Because it is very much important. Okay, and so the milk contained the human alpha lactalbumin and was nutritionally a more balanced product for human babies than natural cow milk. So obviously human babies can be fed with human milk itself, right? Not exactly human milk, but you are giving all the proteins that are there in the human milk. So why not? Why can't we use it? So this is very much important. Now the little details that is over here, children. Cow. Please do not go and make it buffalo there. Okay, and I'm not joking. The questions have come where you have to find the wrong statements. I'll be showing you the questions as well. So there, cow is being mentioned. In 1997, there was a transgenic cow which was being produced, which was Rosie, who was producing high protein milk. It's not high fat milk. It's high protein milk. What is the name of the protein, children? Alpha lactalbumin. Get it? So all this with the biological products. So you are using transgenic animals for the production of biological products now. Okay, that is one thing. Now going ahead, what is next? Vaccine safety. So let's see, what is it? Transgenic mice are being developed for the use for testing the safety of vaccines before they are used on human beings. So again, what I'm going to do is I have the transgenic animal, that is mice. So I'm making mice because 95% of the time they are only being used over here. So what I'm going to do is I'm going to insert vaccine over here and see whether it's actually effective or not. Before putting it into an organism like human being, we put it in transgenic organisms, okay, and we'll get to know what type of behavioral changes or anything are they showing up. Is this something really bad for the body? Anything, you'll get to know about that. Now there's again one very important example that is given over here in your textbook, which you have to read. So transgenic mice are being used to test the safety of polio vaccine. So there is a vaccine that somehow you have to remember. Polio vaccine, children. Polio is caused by a virus. Okay, so this polio vaccine has been given to the mice, making it transgenic mice. Okay, it has been used to test the safety of this particular vaccine. This is important, remember that. Now if successful and found to be reliable, they could replace the use of monkeys to test the safety of batches of the vaccine. So usually monkeys are being used because monkeys and us, you know how much we have much more common ancestry than mice and us, obviously. Okay, so we are mostly closely related to monkeys, and monkeys have been usually used for testing, but nowadays because of so many ethical issues that have been coming up, there is a drastic shift from monkeys to different organisms, and one such organism has always been mice, which is why I said transgenic animals which are being used priorly nowadays is mice. Okay, works. So vaccine safety is one thing that I want to check. Now there is one more thing very similar to this, that is chemical safety testing. So what do you do in that is that you have the mice, okay, again this time it's kind of very angry because I'm going to do something like that. Over here I am going to put a gene in their DNA which is going to make them more sensitive, to a drug. More sensitive as in, normally they are this much sensitive probably, just imagine they are some amount sensitive to one particular drug. But as of now, I am introducing certain genes, certain things in their body such that they are more sensitive. So that, why am I doing that? So that when I am actually putting the drug over here, I want to see what are the most drastic effects that I could find. Second thing, how much dose of this drug could be prescribed example, if I made this particular organism a little sensitive, for example, only 10% sensitive to some drug, and still when I'm putting this drug, if this organism is showing too many drastic features which are not really good, that means probably either this drug that I am taking is not something that can be used on human race, or probably I have to make sure whatever things are being used over here, I have to bring out certain changes, that with 10% usage of this thing, or basically just a little amount of this particular thing is very toxic, so I have to change the whole concept on which this thing is built. Okay, so basically I want to know the most drastic effect that can be caused because of one particular drug over there, and how much dose of it would be really sufficient for a human being to survive normally. Okay, so this is known as chemical safety testing. So this is known as toxicity or safety testing. The procedure is same as that used for testing toxicity of drugs. Transgenic animals are made that carry genes which make them more sensitive to toxic substances than the non-transgenic animals. So I can find similar mice over there which is not very sensitive to this particular thing. Again, 10% of this thing when I'm putting over in that particular mice, it's not very sensitive. But over here I have made some changes over here such that 10% of something is also bringing out a huge drastic effect, which means probably I should not be using, because just like how mice are different, similarly human beings are also different. Some people might have genes which makes them very sensitive to certain drugs, some people might not have that particular gene. So we have to make something which would be equal for both type of organisms or human beings that are there. Okay, so they are then exposed to that. Okay, so what are we doing? We are basically putting the sensitive gene over there which makes them sensitive. Now they are then exposed to the toxic substances and the effects studied. Toxicity testing in such animals will allow us to obtain results in less time as well. So we saw the use, or basically we understood the role of one particular gene with the help of transgenic animals. We saw that we were able to study diseases because of this, the different treatment methods that we can apply on different organisms. If one particular organism have one particular disease, what gene alteration and things can be done. Okay, then we studied about what, children? We studied about the biological products that we had. You had alpha antitrypsin, which was used to treat emphysema, which is a protein that was being made in human beings and is made in human beings, but the gene of it was put in some other organism for bringing out more of this particular protein. Similarly, there was a transgenic cow which was being made in 1997. Okay, its name was Rosie. She was able to produce high protein milk, and this protein, high protein milk, is a human protein, that is alpha lactalbumin, which is found in human beings. So basically this can be given to human children. Obviously it has more nutritional value. Now this biological products were also used to treat phenylketonuria and cystic fibrosis. One more thing, study of what all disease, children? Cancer, okay, cystic fibrosis, rheumatoid arthritis and Alzheimer's. Fine. And over here insulin-like growth factor was the one that was an example of it. Going ahead. Vaccine safety, for this we used polio vaccine in transgenic mice. Safety testing of polio vaccine was done on mice. Okay. Now chemical safety testing, I told you about this thing where you are introducing a gene which makes them more sensitive to any sort of drug such that the organism will definitely show its effect off. Okay, works. So that is the use of transgenic animals. Now before doing anything in the field of biotechnology, you need, obviously, people, if we are living them on their wheel, they'll go probably testing on all different organisms and making the ecological balance all tuned up. So we do not want that. Hence, we have a committee which is known as Genetic Engineering Approval Committee. What do they do, children? Decisions regarding the validity of GM research and the safety of introducing GM organisms for public services, this is all being approved by them. Okay, Genetic Engineering Approval Committee. So whatever genetic engineering we are doing, they have to be approved. So you know, in China and all, we say that they have made so many bioweapons. So again, if I am using this bioweapons, for example, I am altering certain things over here, certain DNA over here, certain genes over here, such that I am making this organism something that is not really good and introducing them into the environment. Don't you think this particular organism can cause the ruckus and kill all the human beings, or probably transform into something else which makes all the more lots and lots of difficulties? So obviously there should be someone who says that, hey, this particular product which is being produced, it's not something that can be used in today's time and it has to be discarded. So the genetic engineering approval committee basically is dependent on that. They make the decisions regarding the validity of all the GM research. For example, should you be doing, so anyone before they start off research, obviously they have to show this is what I'm going to do. So the person who will be there in this committee will approve whether this particular thing can be done here or not. Okay, and also once it is being made, can they introduce this particular organism that is being made in the use for the public services, as in for production of medicines or drugs or whatever things are there. Works? So the thing you have to remember is the full form. The full form is asked. Genetic Engineering Approval Committee. Simple as that. Now, biopiracy. So what happened is that, okay, we made all the things transgenic and things like that, a lot of things happened, but along with it, there were little bad things also happening with us, that is Indians. What was that? Let's see. So over here, let's read. The diversity of rice in India is one of the richest in the world. You know, right? We are the people who make so many different varieties of rice. Basmati rice is distinct for its unique aroma and flavor, and 27 documented varieties of basmati are grown in India. How many? 27. 27 of different varieties of basmati itself are being grown in India. Now, there is a reference to basmati in ancient texts, folklore and poetry, as it has been grown for centuries. So about it lots, and we can see it in lots of places where about this particular thing it has been introduced. Now in 1997, an American company got patent rights on basmati rice through the US Patent and Trademark Office. Okay, in 1997, an American company in the same year the Rose was built, okay, they got patent on basmati rice. Patent on basmati rice, which was produced by, what do you mean by patent? Basically it says that I have the right to produce this thing, no one else has the right, I am taking the patent. This is what you mean by patent. For example, you made something and you say I have the patent right for it, that means no one is allowed to use it any further without my permission. Okay, so or produce that thing without my permission. So in 1997, an American company got patent rights on basmati rice through a US Patent and Trademark Office. This allowed the company to sell a new variety of basmati in the US and abroad. This new variety of basmati had actually been derived from Indian farmers' varieties. So this new varieties that they were talking about was actually taken from the Indian farmers itself, which is kind of very bad, right? You should not be doing that. Now, Indian basmati was crossed with semi-dwarf varieties and claimed as an invention or a novelty. So what was taken is that the Indian basmati which was being used, it was crossed with a semi-dwarf variety of basmati, and then a new variety was being introduced, which is what the American company got its patency in the year 1997. Anyways, the Indian basmati was taken from India, so how can you take the patency right? That is not really fair, right? So even though you crossed it, but initial thing you got it from somewhere else, and probably in India, which is a country which is rich in producing too much amount of rice, they can also do it. So how can you take the patency for that? That was the question. So the patent extends to function equivalence, implying that other people selling basmati rice could be restricted by the patent. So what is the problem of this patent? That anyone else who is trying to sell this basmati rice could have serious repercussions. They could be, they could have to go to court, they could be jailed, anything could happen if someone is using patency. Now several attempts have also been made to patent uses, products and processes based on Indian traditional and herbal medicines such as turmeric and neem. So apart from the basmati rice, turmeric and neem were also tried to be made patent. Now that is something scary, because again that is something that we find very easily in India. Okay, now if someone is having patency for it, obviously production of all these things which is associated with turmeric and neem will be stopping, and you will not have any rights to produce this in your own country, which is very rich in production of all these things. You get it? So this was the problem that was being caused. Now if we are not vigilant and we do not immediately counter these patent applications, other countries may encash on our rich legacy and we may not be able to do anything about it. So if we are not aware about this patency thing, so anyone, when they are saying that I have the patency for this, they provide the submission form and things like that. If we are not aware about what is happening in this world, probably we won't be able to say that, hey, this patency form, whatever you have put forward cannot be, cannot be completed, cannot be of use, because obviously we are a country which is rich in this and we have to use its product efficiently. So you cannot have patent rights on this. Okay. Now this is what, if you understand this, you will also understand what do you mean by biopiracy. So biopiracy is a term used to refer to the use of bioresources by multinational companies and other organizations without proper authorization from the countries and the people concerned, without compensatory payment. So for example, this company itself, okay, let it not be America, any other X company, okay, any other country which is there, or any other company which is there. Okay, so what they are doing, Indian company also you can take. The farmers, from farmers you are taking the rice and you are selling it without giving them payment. Okay, without telling them anything, which are there, you are not sharing your profit with them, then that is a problem. You are a multinational company or you are some organization and you are not having proper authority over anything. You are just taking that particular rice and selling it with the help of your name somewhere, and you are not giving compensatory payment to the people who are associated with it. Okay, then that means that is biopiracy. Okay, so biopiracy is a term used to refer to the use of, whenever I am using like this, I am the multinational company and I am using things like that and I am not giving proper authorization, okay, from any other company, country. For example, I am an American or an Australian or someone, okay, and I am using Indian products, but I am not giving due credit to India for that. Okay, I do not have proper authorization. I am taking things and going without with the help of illegal means. That means I am doing biopiracy. Now I am also not paying them. That's really bad. Okay, this is what you mean by biopiracy. Now questions have come on biopiracy as well. So they have given a definition for biopiracy and they have asked what do you mean by that? So you should be knowing what you mean by biopiracy. If you do not have any idea, you won't be able to solve the questions as well. So this is what we had to learn today, my dear children. Okay, we studied the use of transgenic animals. We know 95% of the transgenic animals is mice, and we studied about GEAC, that is Genetic Engineering Approval Committee. We also spoke about biopiracy, which is again very much important. And now we are going to see some questions. I want you to solve these questions and do tell me in the comment section or in the chat box, and then later on I'll be solving the question towards the end. So are you ready? Let's see it. First question. Read the questions really carefully, okay? Come on, brain cells, work. Okay, so read the following 4 statements about certain mistakes into with them. Okay, so there are 2 statements which has certain mistakes. You have to find that. The first transgenic buffalo raw seed produced milk which was human alpha-lactalbumin enriched. Restriction enzymes are used in isolation of DNA from other macromolecules. Restriction enzymes, they are saying that it is used for isolating the DNA from other macromolecules, as in, what do you mean by that? Proteins, okay, RNA, carbs, that is what do you mean by macromolecules. So basically they are saying that restriction enzymes are being used to separate DNA from all these things that you are seeing over here. Okay, next thing, downstream processing is one of the steps of our DNA technology. Downstream processing we studied in the start of this chapter, also in the previous chapter. Okay, downstream processing as in when you have made something, okay, after that you have a transgenic animal that is going to produce a protein. Now you are separating the protein out from all the other waste and everything that is being produced there. Okay, you are purifying it, you are preserving it. That is what you mean by downstream processing. Okay, and they are saying the downstream processing is one step of recombinant DNA technology, that is genetic engineering. Is that one step of recombinant DNA technology? You have to tell me. Okay, disarmed pathogen vectors are also used in the transfer of our DNA into the host. What do you mean by disarmed pathogen vectors, children? Basically, for example, there is a pathogen, okay, and over here this is a plasmid, and then the plasmid, there is a protein, okay, there's a gene that is coding for a protein which is going to cause trouble to a plant. So when this organism is going to affect the plant, basically this plant will get diseased because of this particular thing. Now disarmed pathogen means I am removing this particular portion, okay, of the gene which is going to code for something really bad. Instead of that, I am going to put my gene of interest, then I am going to put it in the plant. This is a disarmed vector. This is what? Disarmed, as in the armed as in something that protects it, right? So obviously I am removing that part which was causing all the trouble. So it is now not something that can cause trouble over here. So that is why I call it disarmed vector. Why a vector? Because it is going to carry the gene of interest over here. So disarmed pathogen vectors are also used for transfer of our DNA, recombinant DNA, into the host. Is it true or false? This thing that you see is recombinant DNA. So now these questions, or the questions that you see over here, has so much resemblance to the questions from your biotechnology principle and processes. It's a mix of that. Now this question was mentioned in this particular chapter. So if you are not aware about the biotechnology principle and process, it becomes a lot more difficult to understand biotechnology and its application. So if you have not watched the video for that particular chapter, go watch it. Even though I explained a bit over here, but still it's a really nice chapter. I have tried to really explain it well. So please go and watch it. So now among them, try to find the statement which have the mistakes. There are two statements which have the mistake. You will be able to find it really well. So I've explained the things, you tell me the answers. Now, second, genetic engineering has been successfully used for producing what? Transgenic mice for testing safety of polio vaccine before use in humans. Transgenic models for studying new treatments for certain cardiac diseases. Transgenic cow which produces high fat milk for making ghee. Animals like bull for farm work as they have superpower. 2010 question from NEET. Okay, try it out. Now next question that we have. Production of human protein in bacteria by genetic engineering is possible. So we are producing human protein in bacteria, and it is possible because the human chromosome can replicate in bacterial cell. The mechanism of gene regulation is identical in humans and bacteria. Bacterial cell can carry out the RNA splicing reactions. The genetic code is universal. What is the answer for this? Now this is a question which has high resemblance to this chapter, that is molecular basis of inheritance. Okay, why do you think that basically a bacteria which is a prokaryote, a bacteria which is a prokaryote is able to produce a eukaryotic protein? Eukaryotic protein. We humans are eukaryotes. How can they produce our protein? So what is the perfect answer for this? If you are someone who were there with me throughout the entire process, you would be able to answer this question. Even if you are not able to answer, it's totally fine. Understand, try to figure things out. Okay, just cool and try to figure things out. Read through the options, you will be able to get the answer. Now next question that we have, fourth. In India, the organization responsible for assessing the safety of introducing genetically modified organisms for public use is, what is that organization's name, which is telling that okay, now you can probably use this GM product that we have made. So Indian Council of Medical Research, direct question, okay. Council for Scientific and Industrial Research, CSIR, Research Committee on Genetic Manipulation, RCGM, Genetic Engineering Approval Committee, GEAC. This came in NEET 2019, a very direct question and a very easy question as well. Okay. Now next question that we have, fifth question. Use of bioresources by multinational companies and organizations without authorization from the concerned country and its people is called as bio-infringement, bio-piracy, biodegradation, bio-exploitation. Now if you are someone who have not studied this chapter properly, if you are someone who do not know about bio-piracy, the answer that you probably give is bio-exploitation. But this is not the answer, you know the answer. It's a very direct question. Again, this came in NEET 2018. So you see two questions, very direct questions from your NCERT from this particular chapter in NEET 2018 itself. There are so many other questions, I have not included all of them, but yes, you see the irony over here. If you study this chapter, you are getting these marks as well. So please do that. Now let's solve. I hope that you have solved from your end. Let me try to solve it for you and you check the answers now. So first question, read the following four statements about certain and tell the mistakes. Tell the statement which has the mistakes. Okay, the first, transgenic buffalo, it's not buffalo children, it's rice, sorry, cow. Okay, it's cow and the name is Rosie. So that was being used for the production of human lactalbumin. Now restriction enzymes are used in isolation of DNA from other macromolecules. No children. Why do you need restriction enzymes? For example, if this is the DNA and I have the gene of interest over here. I am going to cut the gene of interest with the help of enzymes which are known as restriction enzymes. They are going to make a cut over here, they are going to make a cut over here, and then I can use this gene of interest and put it somewhere else. That is why I need restriction enzyme. So it is not used for isolation of DNA from other macromolecules. Other macromolecules as in proteins, fats, whatever I have, other macromolecules are not separating this thing from this thing with the help of restriction enzymes. No, for them, for doing that I have other enzymes. For doing that I have other enzymes. I'll probably treat with proteases to break down all proteins. I'll put lipases to break down all lipids, but I won't use DNases which would break down my DNA. So DNA is kind of safe, rest everything will be killed. So this statement, again, this is wrong. Now this question is a mix of two chapters, that is biotechnology principle and processes, and this. So this is wrong. It is not used for isolation of DNA from other macromolecules. So A and B are wrong, rest all the things I explained it also previously, it's correct. Downstream processing, yes, it's a step of our DNA technology. Disarm pathogen, yes, they are also used as a vector which carry the recombinant DNA. So A and B becomes wrong. Okay, next thing, genetic engineering has been successfully used for producing what, children? Transgenic mice for testing safety of polio vaccine before use in humans. So this is a direct question from your NCERT and you have to give answer like this. You know it was transgenic mice which we were using for polio vaccine testing, not anything else. So this becomes the correct answer. Going ahead, the next question. Production of human protein in bacteria by genetic engineering is possible because how can you produce a eukaryotic protein in a prokaryotic organism, children? Because the genetic code is universal. The genetic code that you see, it is universal. As in, what do you mean by genetic code? I told you all the organisms know they have their genetic material DNA. Okay, they transcribe it into mRNA. Now this mRNA is used to make proteins. Now mRNA, which is like this, it will have codes like UAA, UCC, whatever. Different codes are there. So these code, okay, this code codes for some amino acid, this codes for some other amino acid. Now these things that you see over here, okay, all of them will be having this and all of them will be coding for amino acid. This is same for all. So the genetic code that you see over here, it's universal for prokaryotes and eukaryotes. Now if you are producing the DNA, you are taking the DNA of a eukaryote, you are putting in there. They will produce mRNA, they are going to produce something like this, and they are going to produce the protein as well, because the genetic code is anyway same. I am going to make the same amino acid, the amino acid will come together, make the same protein. So this is a question from your molecular basis of inheritance, okay, and it has been put in this particular chapter. So you see, as I said, if you do not know about the basics in there, you won't be able to solve in here as well. All the chapters that you see are somehow interconnected. Okay, this came in NEET 2005. Now India, the organization responsible for assessing the safety of introducing genetically modified organism for public use is GAAC. It is not appraisal, it is approval. Okay, this came in NEET 2018. Direct question. One more question was there. Yes, the use of bioresources by multinational companies and organizations without authorization from the concerned country and its people is known as biopiracy. This will be known as biopiracy. This came in NEET 2018. Works? Cool? Now, one cute little homework for my cute little cuties, and tell me one more thing. If you want a one-shot video, one-shot revision for this particular chapter as well, just like how I made it for biotechnology principle and processes, you can let me know in the comments. Okay, only if you tell me, then only I'll be doing it. Cool? Now, okay, so a new variety of rice was patented by a foreign company, though such varieties have been present in India for a long time. This is related to what? L67, Sharbati Sonora, Lerma, Rojo, Basmati. NEET 2018 question. Again 2018. The three questions that I showed you here itself. Okay, so you see the number of questions that are coming from this chapter, especially from this topic. In one particular year, I got 3 questions from the same topic. Okay, so please try to solve it and do let me know the answers in the comment section. I do not see that vigor in the comment section as of now. Where are you all? Biology people, come on. If you are not showing this motivation, probably we would not think of going forward with this thing. If you are not getting utilization of this, do let me know in the comment section that, ma'am, please, we have to work on this. This is not working somehow. Or tell at least good things about this so that we have the motivation to keep going. Yeah? Now, If you think that this particular video was very helpful for you, then do not forget to subscribe to the channel, and also not forget to be happy and read your NCERT. It's very much important for biology. Okay, so bye bye children, take care, wishing you all the best for your preparations.